All right, so now we're going to switch to talking about lipids. The lipids don't really fit our chart as neatly as some of the other things because they're not macromolecules. They're different because they don't have monomers, they don't form polymers, okay? So the unifying characteristics that make lipids lipids are that they don't form polymers, and the other major characteristic that they have is that they're hydrophobic, okay? They don't mix well with water. So we're going to look at a couple of different examples of lipids. The lipids we're going to talk about include fats, phospholipids, and steroids or steroid hormones. The first one we're going to talk about are the fats. Okay, so fats are what we normally think of when we think about lipids. Fats are lipids. Not all lipids are fats, right? There are other fats, or excuse me, there are other lipids, phospholipids and steroids. So a typical fat is a triglyceride. Triglyceride is made of a glycerol, 3-carbon sugar alcohol that has hydroxyl groups coming off here and hydrogens filling in the rest of the bonds for the carbons. Okay. And on each of those hydroxyl groups is a place where a fatty acid can be joined to the glycerol molecule. So I'm not going to draw the whole fatty acid here, but a fatty acid is a chain of carbons that have a carboxyl group on the end. Okay? And so just like we saw with our saccharides being joined together, we're going to join our fatty acids to the glycerol with a dehydration reaction. So the hydroxyl group from one is going to react with the hydrogen from the other. They're going to leave and form water, and a new bond is going to be formed. The new bond that's formed is an ester linkage, but I want to point out that this is not a polymer. We joined together two dissimilar things. We joined glycerol to a fatty acid. So we didn't make a polymer, we joined these two different pieces together. So the same reaction can be used to add fatty acids to each of the carbons in this triglyceride. So we have our 3-carbon glycerol molecule that has 3 fatty acids attached to it. So our specific example of a particular fat is a triglyceride. But what those fatty acids are can actually vary. There can be different kinds of fatty acids attached to our glycerol molecule. And the nature of those fatty acids is going to change the nature of the fat. If you zoom in on one of those fatty acid chains, what they are a chain of carbons hooked up to one another. Each carbon has 4 valence electrons. It has room for 4 covalent bonds. So if we start with this carbon in the middle, it's attached by one bond to the carbon next to it and another bond to the carbon next to it, and then the remaining bonds are filled in by hydrogens. And when this is true for every carbon in the chain, every carbon is saturated with the maximum number of hydrogens that could be attached. So if our carbons are each singly bound to their neighbor and bound to hydrogens, they're saturated with hydrogens, that makes a saturated fatty acid. But carbon can also form double bonds. So let's imagine that we have a double bond here instead of a single bond. Now this carbon's participating in one bond with this carbon over here and 2 bonds with the carbon over here, so it only has room for 1 hydrogen. Same thing over here. This carbon's already participating in 3 covalent bonds. It only has room for 1 more. And when you have the hydrogens on the same side, it actually causes the fat to bend into this kinked shape. Okay? And so these bent fatty acids are called cis fatty acids. Okay? They're cis unsaturated fatty acids because they have a lower ratio of hydrogens to carbon. They have fewer hydrogens because of the presence of double bonds. So cis unsaturated fatty acids, because they have this bent shape, can't pack as closely to each other. So here in the slides it shows this kinked or crooked one. Saturated fats, since they're linear, can pack closely together and they form solids at room temperature. So when you think of butter or bacon or the big slab of fat on the top of a pork roast or the veins of fat in a steak, those are saturated fats. Saturated fats are typically made by animals and they're typically solid at room temperature. Unsaturated fats that have a double bond, cis unsaturated fats, are kinked, and so they push the neighboring fats further away, and that allows them to remain fluid at room temperature. So unsaturated fats are things like vegetable oil, olive oil, and they're typically made by plants. But we have this one other example over here, which is a trans fat. What happens when you either take a saturated fat and you remove the hydrogens and you hydrogenate it, the shape of the bond that's formed is different. Instead of putting the hydrogens on the same side, they end up across or opposite from one another. So this makes a trans fat. And when these hydrogens are opposite of each other, it allows the molecule to line up straight. It doesn't bend. So even though it has a double bond, even though it is an unsaturated fat, it has the same shape as a saturated fat. And so these trans fats can pack together and form solids. They're also preferred for packaged food because they're shelf stable. They don't spoil as quickly as natural saturated fats. And so they're in processed food. But they act like saturated fats in our body because they can pack closely together and contribute to atherosclerosis and heart disease. These trans fats had to be separately labeled in nutrition packaging information. However, if you have a package of food that says it has 0 grams of trans fats, but the ingredients label says that it contains partially hydrogenated vegetable oil or soybean oil, it has trans fats in it. They're just allowed to round it down if it's less than 0.5 grams. So if it says hydrogenated vegetable oil, it has trans fats. The function of fats is for long-term energy storage. And also insulation, padding of body organs, and other fats that also contribute to the structure of animals and all organisms really include phospholipids. So phospholipids are built in a very similar way as our triglycerides. They have a 3-carbon glycerol molecule. On 2 of those hydroxyl groups, fatty acids get attached. But on the 3rd one, instead of adding a fatty acid, a charged head group that contains phosphate is attached. Okay. And so there's different functional groups that can be in these polar head groups, but the important thing is that they all contain phosphate. And that phosphate group ionizes in water. It's negatively charged. Okay. So now we have part of the molecule that is made of fatty acids that are very hydrophobic, but we have this head group that is hydrophilic because of the phosphate group that mixes well with water. Okay? And so what happens if you take a molecule that has both hydrophobic parts and hydrophilic parts and mix them in water, all of the hydrophobic parts are going to bunch together to get away from the water, and all the hydrophilic parts are going to mix with water. So if we do this with a simpler molecule that has a phosphate group but only 1 fatty acid tail, what happens is all the fatty acid tails try to bunch together to get away from water, and all the polar head groups point out and mix with the water. And this is how phosphate-based detergents work. The hydrophobic tails surround oil droplets in your dishes or your laundry and separate them from whatever they're soiling, and the polar head groups mix with water by hydrogen bonds and separate the dirt and the oil from whatever they're being washed off of. Now, phospholipids, since they have 2 tails, they don't form these bubbles or micelles that go all the way around. Since they have 2 tails, the same thing is trying to happen. The hydrophobic tails bunch together to get away from water, but before they can go all the way around and make a bubble, more phospholipids fill in on the other side. And then eventually these curve all the way around and make a big bubble. And it's that shape, this double layer of phospholipids, that makes up the membrane of cells. This is the phospholipid bilayer. And all cells, from bacteria to humans and everything in between, have a cell membrane that is arranged like this. All right, so we have one more class. Oh, so the vocab words that go along with this. So hydrophilic is anything that loves water, that mixes with water, and that's going to be things that are polar or ionic. And so our phosphate group here and also the amine group in this head group make the top half of our phospholipid hydrophilic. Hydrophobic molecules are non-polar. They don't mix well with water. And so our hydrocarbon chains and the fatty acid tails that are just non-polar bonds between carbon and carbon or carbon and hydrogen are hydrophobic. So when you have a molecule like a phospholipid that has both a hydrophilic head group and a hydrophobic tail, that makes an amphipathic molecule. And those are all vocab words you guys should be familiar with. Our last class of lipids are the steroids or the sterols. So these look a lot different than the other phospholipids that we've talked about so far. Instead of a straight hydrocarbon chain, those hydrocarbons get folded on top of each other to make these 4 fused rings. Now, we've got hexagons here, but just because they're hexagons doesn't mean they're sugars. They're not sugars at all, okay? So this structure is not shorthand. So if we draw our fused rings, so first of all, there are 4 rings that are fused together, okay? 3 of them are hexagons and 1's a pentagon, okay? And then there are these hydrocarbon tails that come off of them, okay? So in each of these hexagons, there's a carbon at each point, okay? And the illustration isn't leaving much out. Off of each of these carbons, there's just hydrogens for the most part. If there's something other than hydrogen, then they draw that hydroxyl group that is distinct. Okay? So each point is a carbon and each point is full of as many hydrogens as it has room for. So they're still hydrophobic. Okay? The function of steroids is they play a structural role. They're found in our plasma membranes, but they are also used for signaling. These hydrophobic molecules can be absorbed directly by cells. They can squeeze through the phospholipid membrane and then provide information to the cell to change gene expression. And I want you to appreciate how subtle the differences are between the different steroid hormones for males and females. So cholesterol is a structural component of our cell membranes, and this hydrocarbon tail gets clipped and replaced with a hydroxyl group. And this hydroxyl group forms a carbonyl oxygen with the carbon here in testosterone. Estrogen's very subtly different. It's just changing one bond, basically. So it's replacing this carbonyl oxygen with a hydroxyl group and removing some of the hydrogens from this ring. So subtle molecular changes make the difference between males and females. Looking back at your study guide, for lipids, they don't have monomers and they don't form polymers. Okay, so those are not relevant. But to form a single lipid, you do a dehydration reaction between glycerol and the fatty acid tails to form an ester linkage. So examples that we went through were fats. Specifically, we gave the example of a triglyceride, which is used for energy storage, phospholipids, which have the structural role in our cell membranes, and steroids, which have the role of cell signaling. One of the major unifying characteristics for lipids is that they're hydrophobic. They don't mix well with water. So you don't have to be able to draw a cholesterol molecule, but you do need to be able to recognize cholesterol or a phospholipid or a triglyceride and know that those are lipids.